Okay, assalamualaikum guys. Today we're gonna go into our third chapter, Introduction to Genetic Engineering in Biotechnology. So what is genetic engineering? So genetic engineering is a part of biotechnology. So biotechnology is the use of artificial methods to modify the genetic material of living organisms or cells to produce novel compounds or to perform new functions. So we have been using biotechnology, as you have learned, to improve livestock crops since the beginning of agriculture through selective breeding. Now, since the discovery of the DNA structure in 1953 and the development of tools and methods to manipulate the DNA in the 70s, biotechnology has become synonymous with the manipulation of organisms' DNA at the molecular level. Now, so using the technology or the DNA technology, aka biotechnology methods to modify an organism's DNA to achieve desirable traits is called genetic engineering. So as you can see, there are many terms that can represent genetic engineering. Some people call it gene manipulation, some call it gene cloning, recombinant DNA technology, or for the punya short form is small r DNA, and then genetic modification, and some refer it to as new genetics. So by definition, genetic engineering is the deliberate, meaning intentional manipulation of DNA using techniques in the laboratory to alter genes, to change genes in organism. Even if the organism being altered are not microbes, the substances and the techniques used are often taken from microbes and adapted for use in more complex organisms. Now, if you look into this diagram, this is a bacterial cell with its chromosome inside and a plasmid inside. So you know that a bacteria, they can have extra chromosomal DNA, which we call as plasmid. Ini the whole genome dia. So the plasmid is something that we, the researchers, can manipulate. You can take out the plasmid out from the cell and insert a gene from some or other organism and combine it together into the plasmid, creating recombinant DNA. So you know recombinant DNA is a mixture of this original DNA plus a sequence of DNA from other organism. And then what you do is that you're going to transform back the plasmid into the bacterial cell. Now, ni original dia. You see tak ada sequence warna biru? In the recombinant bacteria, meaning something that you have changed, or bacterial cells yang dah take up this new plasmid, they have this new sequence in the plasmid. And then when they divide, they will clone together the new plasmid. So anak-anak dia semua ada all that new DNA. Okay, so this is used technique of cloning new genes into the plasmid of bacteria, which act as a host, is used in many fields. In the medical field, gene therapy, making new protein, insulin for example, creating transgenic plants. You use the genes to alter bacteria for cleaning up toxic waste. Okay, so that's dia punya kegunaan, mengapa, why we actually use genetic engineering or recombinant DNA technology. why we alter genes in organisms.